All right, guys, in this video, we're going to talk about mitosis. So starting with our interphase right here, one thing that you should notice about the interphase is that you cannot see the X-shaped duplicated chromosomes, right? Even if the duplicated chromosomes are already made during the S phase, the S phase, and then during the G2 phase, our chromosomes are also duplicated, or DNA is already duplicated, you can't actually see the X shape, right? Because they're not condensed into the X shape yet. We don't need DNA to be condensed into the X shape unless we're ready to pull them apart. And we're doing the pulling apart part during mitosis. So let's look at how mitosis works. I'm going to draw on the board, which is basically the same thing as prophase, but just in case you want to follow along. So we start with, yeah, that's fine. We start with our prophase. You should know that you can try to remember it as PMAT plus C. So PMAT C. However you want to remember it, it's fine. But you should know the sequence of each step, okay? Starting with mitosis, first step, prophase. Prophase, if we were to have two chromosomes, by this point, they should all be duplicated chromosomes. So you should have a nucleus, and there should be 4 X shapes in there if we started with 4 chromosomes, right? So before this happened, before the S phase, during G1 phase, you would have a cell, and you would have 4 unduplicated chromosomes like that. See, unduplicated, duplicated. All right, so this is our prophase. During prophase, the nuclear membrane is already broken down a little bit, so you can draw it as this. But you can still see the nucleus is still there. Okay, so that's prophase. The next one is called metaphase. So metaphase is special because the sister chromatids line up at the metaphase plate. Again, sister chromatids line up at the metaphase plate. All right, so let's draw a cell. The metaphase plate is kind of an imaginary region. It isn't actually a real plate. It's just where the chromosomes line up. So if we were to have 4 chromosomes to start with, 4 duplicated chromosomes, they're all going to line up in the middle of the cell. This is going to look like this. 1, 2, 3, 4. So the place that they're all lined up, that is called the metaphase plate. And as you can see, we have individual chromosomes lined up. There's no pairing of anything. So you would say sister chromatids lined up and not homologous chromosomes. There's no homologous chromosome at all in mitosis. This is very important. There's no homologous chromosomes during mitosis. Right? There's no pairing up. So we have sister chromatids lined up. And then we have something called centrosomes. So they're kind of a T-shape. Centrosomes does a really important job. What it does is that it makes these lines, and they're called spindle fibers. 3, 4. So these spindle fibers attach to the sister chromatids. And what it does is that it will pull the sister chromatids apart from each other. So if your two sister If your two sister chromatids look like this, the spindle fibers will pull them apart so that now you will have 2 separate DNA instead of, 2 separate DNA and 2 separate chromosomes instead of 1 chromosome and 2 DNA attached to it. So that's our metaphase. Let's label it. The next step after metaphase, PMAT. Now we're on to A, so anaphase. In this case, your cell should already be a little bit lengthier than it was before, so it should look like this. We still have our centrosomes that make these spindle fibers, but now the sister chromatids are pulled apart. See? They're all pulled apart like that. All right? So now, here's the difference. Here we have 4, Here we have 4 chromosomes, 8 DNA, because each chromosome is duplicated, right? However, right here we have 8 chromosomes, but still 8 DNA. We didn't do anything special to cut the DNA number into half or anything else. But we have 8 chromosomes now because each sister chromatid had already separated from each other. That's the main event that happens during anaphase. Sister chromatids actually separate. Now we're on to telophase. telophase looks pretty similar to anaphase, except now our cell is even longer than it was before. It shouldn't, this is not very good drawing. So now this is more like it. Here we have telophase. If their cell is even lengthier, and you still have your centrosomes, we still have our spindle fibers. But the special thing that's happening is that we have nucleus starting to form around, we have these two nucleus starting to form around the DNA now, right? So now on to our last step. Now here's the moment of truth. We have cytokinesis, where now finally your cells are going to be, so these cell membranes are going to kind of pinch in. It's kind of like a drawstring in the middle of the cell. Now you have two different cells. They each have its own nucleus. They each have 4 chromosomes. So if you trace this entire process, we started with a cell with 4 chromosomes, 1 nucleus, right, unduplicated. And then during the interphase, things are duplicated. The one thing that you want to make sure to know that we talk about prophase, metaphase, anaphase, telophase. During mitosis, this actual cell division process is when you can actually see the chromosome shape, right? So this right here really would look like this. You can't tell the DNA apart from each other until the prophase, okay? But this is here to show you that we started off with 4 DNA. And then at the end of our PMAT, this is cytokinesis. Now we have 2 different cells, 2 daughter cells that are the exact same copy as our parent cell. They each have 4 chromosomes. The chromosomes aren't duplicated. Done. All right, let's take a look at our review questions, see if there's anything that we didn't cover. Don't think so. If you see anything that you can't figure out on your own, please come talk to me. I'll be at the library in the morning. Bye.